So we're shifting fields completely now. So I'm a PhD student at ISD Austria in the group of Gaia Novarino. And as already Matteo said, I'm working on a large neutral amino acid transporter and its role in neurodevelopment and in neurodevelopmental disorders such as ASD and microcephaly. I'm working on a gene called Slc7a5 that encodes a large neutral amino acid transporter, and this is shown here in light blue. And this transporter forms a heterodimer with another protein, and this heterodimer is then referred to as LAT1. And LAT1 catalyzes a large neutral amino acid flux into different tissues, and among their substrates are also these essential branch chain amino acids. And it catalyzes this flux in a sodium and pH independent manner. So it's highly expressed in different tissues that have a high amino acid demand. Among them is, for example, the brain, which we are mainly interested in. So it's highly expressed in neuroprogenitor cells and in neurons. So why are we exactly interested in this gene? So a few years ago, we identified a subset of patients that harbor homozygous missense mutation in the Slc7a5 gene. All of these patients present with severe motor deficits. They are on the autism spectrum and they have severe microcephaly. So what we are now, in my PhD project, I'm mainly interested in the microcephalic phenotype of these patients. So we're trying to figure out which role Slc7a5 plays in neurodevelopment and how a loss of this transport is causing such a strong neurodevelopmental defect. But before I start, I just want to clarify some terms. So what is microcephaly? So in fact, the patient is diagnosed with microcephaly if the head circumference is 2 standard deviations smaller than average. And microcephaly can be divided into 2 subtypes. On one hand, you can find so-called primary microcephaly, meaning children are born with a smaller brain size, and this is usually due to defects during embryonic development, so defects in neuroprogenitor cells or in neuronal migration. However, there is also this so-called secondary microcephaly, meaning children are born with a normal brain size and start to develop microcephaly after birth due to mainly defects in processes involved in neuronal refinement, such as pruning or myelination. And of course, an increase in cell death of neurons or glia can, for example, contribute to this defect. So how are we studying microcephaly in the Novarino lab? I'm working with a conditional knockout mouse model of Slc7a5, where we're deleting the transporter in the developing cortex and hippocampus, more specifically in radial glia progenitor cells and their offspring, so excitatory cortical neurons and astrocytes, at a very early developmental time window, so at E11.5. What was quite curious to see was when, although we're deleting this transporter so early in development, these mice are actually born with a normal brain size. However, they start to develop postnatal microcephaly. So this you can nicely appreciate here in this plot, where you can see embryonic time points and early postnatal days after birth, and this is the brain weight, and you can start seeing that there is a difference in the brain weight starting to arise at around day 2 or day 3 after birth, which then gets really obvious at day 5 to day 6. However, after day 10, this difference stays constant, meaning what we're seeing and the effect that the loss of Slc7a5 has seems to be restricted to this very critical period after birth between P2 and P10. And this actually has been shown in neuron development to be a crucial time window where neuronal refinement is taking place. So the first thing that we of course did when we saw these changes in in brain size, we looked at general brain morphology. What you can see here is that these mice show a severe reduction in cortical thickness by around 40%. Looking at the cortical layering, you can see that especially the upper layers, so layer 2, 3 of the cortex, are significantly affected. So their thickness is severely reduced, whereas the lower layers here are affected, but is not as strong as in the upper layers, indicating that the layer 2, 3 pyramidal neurons seem to be more susceptible to the loss of Slc7a5. seeing such a severe phenotype, we of course, first of all, wanted to know what is happening to these neurons. So the first thing that I was checking is apoptosis. So what if these neurons just die? And indeed, this is what we're seeing. So I did immunoblots and immunostainings for cleaved caspase, which is like one of the main apoptotic markers. And this is a time course from P0, so birth, until adulthood. And what you can see is that we see a significant increase in apoptotic activity, again, during this very distinct time window where we're also seeing the microcephalic phenotype arising. What was quite curious to see is, so for people that are familiar with the literature on this postnatal period of neuronal refinement, this peak kind of is quite well known, especially regarding programmed cell death and neuronal refinement, because it has been shown in literature that in cortical development, so murine, but also in human cortical development, exactly during this time window where we are seeing the increase in apoptosis, so between P2 and P10, a wave of endogenous cell death is moving through the cortex, eliminating different cell populations due to, or to ensure a properly functioning network. So what has been shown by the marine lab is that actually between P2 and P5, you have first of all a wave of apoptosis hitting pyramidal excitatory neurons, and later between P5 and P10, exactly the window where we are also seeing the increased apoptosis, you see a wave of apoptosis hitting the inhibitory neurons, making sure that the cell numbers are adjusted again and that the ratios between the cell types are constant and a proper network is formed. So what has been shown, the factor which is modulating this cell death wave is actually neuronal activity. So neurons which are less active tend to undergo apoptosis, whereas neurons that are more active remain integrated in the system. So this was quite interesting for us to see, especially since we also see the wave of apoptosis in our mutant mice exactly during this time window, and we wondered if there might be a connection between a large neutral amino acid transporter and neuronal activity. And so we decided to do some electrophysiology. So this was done by a postdoc in our lab, Bernadette. She used current clamp recordings in layer 2-3 pyramidal neurons in acute slices exactly during this time window where we see the increased apoptosis. And indeed, what we find is that these pyramidal neurons in the mutant mice show a significant reduction in their firing rate. What was even more interesting to see was if we repeat these recordings at later time points, so during juvenilehood, these difference is again gone between mutant and wild type littermate control. So what this indicates is that we're really seeing a purely developmental phenotype here, and that it seems that Slc7a5 plays a really important role during this critical developmental time window of neuronal refinement. So another result that is nicely underlining this is shown here. So additionally, what I did is I looked at different other cell types which are present in the cortex, in this case, PV positive inhibitory neurons. What I have to mention here is that these PV positive inhibitory neurons are actually not affected by the loss of Slc7a5 due to the creline that we're using. However, what we're seeing is also that the inhibitory neurons are adjusting their numbers, so undergoing cell death, especially in the layers where we also see the majority of the excitatory neurons lost. What this is showing us is that we're indeed exactly hitting this time window that was introduced by the marine lab, showing that the inhibitory neurons as a secondary effect are also starting to die to adjust their numbers accordingly. So due to time reasons, I have to sum it up already. So what I could show you today was that a deletion of Slc7a5 in excitatory forebrain neurons causes postnatal microcephaly. This is due to an increase in cell death during a very distinct developmental time window. What we also see is that during this distinct developmental time window, we're seeing a decrease in firing rate of cortical pyramidal neurons, which might be even the cause why these neurons start to undergo apoptosis. So what remains now for me, the most crucial question I have to answer now, why do we see this reduction in firing rate? So what we're checking now is it due to the loss of the transporter itself, which might be modulating, for example, voltage-gated ion channels, or is it due to the loss of the substrates that the transporter is transporting? So yeah, with this, I would like to thank everyone that is involved in this project, and I'm happy to take some questions.
Thank you, Lisa. You made more than perfect timing, and it was a very interesting talk.
Cool.
So now it's question time, and I would like to start. So you showed that, can you share again your slides?
Yeah, sorry, I'm just here.
No worries. Okay. You showed that slide where you had basically the histological section of the normal and the microcephalic brain. One next, yeah, exactly this one.
Mm-hmm.
So yeah, so the cortical changes that is evident, and you talked about it. What I wanted to ask you, what is known about instead what is happening at the subcortical level and in terms of the white matter connections?
So the thing is that we cannot exclude that something would also happen in subcortical regions. The thing is, the cree-line that we're using is just targeting the cortex, or the developing cortex and hippocampus. So this is something that we would have to check at one point. In parallel, we were breeding also with the nesting cree-line, which is deleting the transporter in the whole brain. However, this is something that we stopped at one point because the pups were not really very healthy, because we were just removing it from the whole brain. Regarding the corpus callosum, so the white matter tract, I was quantifying if there is a reduction in the corpus callosum thickness, for example. We didn't find any. However, I would, and this is on my list, also want to do some stainings or quantifications of MBP, so to check if there is a decrease in myelinations, just to be sure if this is also contributing to the phenotype, of course.
Yeah, sure. And also because in terms of volumes, of course, I imagine that this reduced volume is both if you look just at the cortical and the white matter, right? And also, I imagine that even in this case, that if I understood right, the transporter you are targeting is just for the cortical neurons, but you may have an effect also in white matter, given that it looks shrunk in the picture. Is that right?
Yeah. So this is something that we really have to check more. I mean, also considering that you lose a substantial amount of neurons which are projecting onto the different, so on the second hemispheres, it would be something that we would expect that you have also a reduction in the corpus callosum, for example.
Okay. So while I'm waiting for questions from the other participants or the other panelists, if they are curious about something, I was puzzled by the fact that basically the inactive neurons die while the active ones are still alive. Do you think that maybe it's a naive question, but is it something that you can exploit to react in some way to the pathology? There would be a way to make the inactive neurons more active over time in some critical period to make them survive?
It's a very, very, let's say, nice. So it's an idea that is really, really nice, especially in the part, like in the field where I'm working in. Of course, we're all trying to find some possible ways of treating these disorders. The problem with microcephaly is that you would have to treat already during, in most cases, you have to treat already during embryonic development, which is, of course, always a bit tricky, especially because these children are often diagnosed after they get born. But of course, it would be very, very a nice idea if this would be possible. However, with boosting activity in humans in the brain can be a bit tricky, because of course you can then induce some other effects that are going more into direction seizures, which you also don't want to boost too much. So I think this would be very, very challenging. But of course, if we find the modulator that is actually decreasing or modulating the activity, we can start to plan or think about to modulate this factor and not the activity itself. That would be the idea, of course.
Thank you. And to conclude, would you like to briefly tell us about how we could, how you could extend these results for therapeutic goals? How this could help to solve the issue?
So I didn't have time to show this, but we actually already found that, so Slc7a5 is also expressed at the blood-brain barrier. If you delete it there, then you will get mice that resemble more the phenotypes that I showed you before, like that are more like autistic-like phenotypes, social defects, and so on. So we were able to kind of treat these mice by injecting branched-chain amino acids, so the substrate that Slc7a5 is transporting into the brain. So this worked very, very nicely and was very, very convincing. However, if we're now seeing that the phenotype here is coming from the neurons, this will be very tricky, because injecting the substrate into a cell type itself will be quite challenging. So this is maybe a bit tricky, but there are possibilities how you can approach this.
Okay, thank you very much for the answers. And since it's 45, we made perfect timing.